In this presentation, we will discuss the concept of genetic drift. Genetic drift is one of several mechanisms by which evolution can occur in populations. The other mechanisms include mutation, gene flow, non-random mating, and natural selection. The term genetic drift was coined by the founder of population genetics Sewall Wright in 1929. However, his initial use of the term correlated more with natural selection, and while they can overlap, unlike natural selection, genetic drift doesn't take into account how helpful or harmful an allele is. Genetic drift occurs when random factors impact the frequency of alleles and subsequent generations of a population. Smaller populations are the most susceptible to the effects of genetic drift, as random events, such as bottlenecks or the founder effect, can cause sudden and drastic changes within a population. For example, in one population of fish, 2 are red and 1 is blue, for a total population of 3 fish. This generation mates and their offspring have a 50-50 chance of being red or blue. By chance, they have 3 offspring and all are red. Once the original population dies, the entire population is now red. In another population of fish, 10 are red and 5 are blue, for a total population of 15 fish. This generation mates, and their offspring also have a 50-50 chance of being blue or red. By chance, they have 9 red and 6 blue offspring. Once the original generation dies, the population is now 60% red and 40% blue. As you can see, the first population of 3 fish was more affected by genetic drift in that it resulted in a complete loss of the blue genotype after just 1 generation. Because of the small population, there was a higher statistical chance that this would occur. The larger population of 30 fish was less affected because the chance of all 30 offspring being one color was very statistically improbable. Understanding genetic drift is important in order to understand one way that species have evolved over time.
Next, we will talk about allele frequency, what it is, and why it's important. Allele frequency is the frequency of the 2 forms of a particular allele in a population, and it is important for the calculation of genetic variation of a population. Alleles that occur at a low frequency are usually at a disadvantage in the process of genetic drift. Low-frequency alleles face a higher probability of disappearing from a population than alleles that occur at a higher frequency. Unlike natural selection, genetic drift does not depend on an allele's beneficial or harmful effects. Instead, drift changes allele frequencies purely by chance, as random subsets of individuals are sampled to produce the next generation. Allele frequency can positively and negatively influence the genetics of a population by carrying a gene that increases fitness, or it could carry a genetic disease.
For the next few slides, we're going to be going over the first type of genetic drift, which is known as the bottleneck effect. The bottleneck effect is an event that happens when the size of a population is extremely reduced. This reduction can happen as a result of natural disasters like floods and fires. It can also happen as a result of overhunting or disease that wipes out many in a population. But either way, it leaves a random assortment of survivors afterwards. Was there before the event will be there afterwards, since some species might have been entirely killed off, or a rare genetic variety might have died off within a species. On top of that, since the population is so much smaller, it'll be even more susceptible to genetic drift, which leads to even more alleles to be lost. Lastly, less genetic diversity within a population leads to the offspring being more susceptible and vulnerable to disease. This photo from Khan Academy is a great representation of the bottleneck effect. The marbles represent the individuals in a population. so the marbles in the bottle represent the original population. The marbles are then being poured out of the bottle into a cup. The pouring represents the bottleneck effect, and those in the cup represent the individuals that survived the event. Those that are left in the bottle represent the individuals killed off. As you can see, not all types of marbles were able to be poured out of the bottle. For example, the red marble. No red marbles were poured into the cup. Therefore, that individual was completely killed off, lowering the genetic diversity in the new population. One example of the bottleneck effect is the northern elephant seals. In the 19th century, the northern elephant seals were nearly hunted to extinction due to their oil-rich blubber. The whole population was reduced to only about 100 individuals. Thanks to the United States government stepping in to protect the seals, they were able to bounce back, and there are over 30,000 individual northern elephant seals today. Though it's great they were able to rebound, there is concern for their long-term survival. With them coming from such a small population, there is very low genetic diversity within this new population, which means the species is very susceptible to illness, and it's possible that the species can be entirely wiped out if an illness were to overtake it.
Next, we will be going over the founder effect. Here are the important things we will go over regarding the founder effect. First, we will go over the definition of the founder effect. Next, we will talk about some examples. Then we will talk about the impacts of the founder effect. And finally, we will talk about why it is important to study. So, what is it? The founder effect is defined as the reduced genetic diversity, which results when a population is descended from a small number of colonizing ancestors. In other words, it is when a few individuals from a big population colonize a new area and starts a new smaller population with a different allele frequency than that of the original. In this example, the redbirds make up the majority of the population on the Big Island. As we can see, one redbird and two blackbirds move from the original population to the small isolated island. Future generations will have a higher frequency of blackbirds on the smaller island due to the loss of genetic diversity. So, what are the effects? Some of the positive things that can come out of it include speciation and mutations. Speciation allows for a population to evolve into a new species, which would help increase the overall fitness of the new organism. Additionally, if any bad mutations were to develop, then it would not impact the original population. Some of the negative effects include limited genetic diversity, interbreeding, and the increased chance of genetic diseases. These three things go hand in hand with each other. Due to the small population and gene pool, the organisms are more likely to develop genetic diseases from interbreeding. If the population doesn't get any new settlers to expand the gene pool, then the population could die off. The bottleneck effect can exacerbate the problems as well by decreasing a population that is already small to begin with. So, why is it important to study the founder effect? It is important to study the founder effect for many reasons. One is so that we can find common ancestors in evolutionary biology. Understanding where a species came from is important in evolution. Two is so we can get an idea of where diseases may have come from. There are bacteria that can harm humans who share an ancestor with other bacteria that can harm different types of animals as well. By understanding their evolutionary history and utilizing the founder effect to study bacterial communities, we could get a better understanding on how they work and how they got to where they are in the evolutionary timeline. A third reason has to do with conservation. Because of what we do understand on the impacts of the founder effect, people who work in conservation know there needs to be gene flow within a community or it will not be sustainable in the long run.
What are the long-term effects of genetic drift? Genetic drift has two significant longer-term evolutionary consequences. It can facilitate speciation, which is the creating of a new species, by allowing the accumulation of non-adaptive mutations that can facilitate population subdivision. It also facilitates the movement of a population from a lower fitness plateau to a higher fitness plateau. The amount of population subdivision is expected to increase because of the random losses of alleles that occur in different populations. In addition, random changes in allele frequencies are expected to occur in different populations, and these random changes tend to make populations become differentiated. Finally, small effective population sizes increase the likelihood that mating events will occur between close relatives, leading to an increase in inbreeding and subsequent loss of heterozygosity. To conclude, the importance of genetic drift is that it reduces the amount of genetic variation in a population. So with less genetic variation, there is less for natural selection to work with.